Okay, I've made my buffer. I filled it up to the line with distilled water or tap water. And now I'm going to fill the inner chamber. So the chamber inside the cassette, the place I tested wouldn't leak before. And what you need is enough buffer in the inner chamber so that it covers the short side of your gel. Remember, the gel has a high side and a short side, and the short side should be on the inside of the cassette. You want to add enough that it actually covers that short side of the gel. It'll be most of the way up, but not all the way. You don't want to overfill this so it spills over the sides. Then you take the rest of your buffer and you dump it in the outside of this container. You should notice it says 2 gels and there's a line, 4 gels and a line. It's going to take it up to about the 2 gel location, or the 2 gel line. And if it's not quite, it's okay. You can add a little bit more water to get to that 2 gel line if you have to. But 550 should just about do the trick. So now this thing is ready to run and you need your samples. So you've got your little thing here and you have some tubes. One of them is a tube of blue stuff. One of them is a microcentrifuge tube, and it has a small amount of blue stuff in the bottom as well. Then you have 2 tubes labeled AP and BSA that have whitish powder in them. These are 2 proteins. So here's what you're going to do. First, you have the one labeled PP that has a little drop of blue stuff in the bottom. This is a precision protein standard. Basically what this is a bunch of proteins, I think 6 or 8 of them, that have been pre-dyed, so they're colored, and they're in loading buffer already. That's what this blue stuff is, loading buffer. And when you put these in your gel, they are going to run and show you what different size, how far different sized proteins get on your gel. So this is going to go in one of the wells in your gel. Your 2 protein samples, you're going to want to add water to these. This is what we call a qualitative experiment, not a quantitative experiment. So exact concentrations aren't going to matter. So I'm just going to add some water to this so that I have some protein. Doesn't really matter how much. There we go. Because I just want to have some. And yes, I know I'm denaturing my protein. Don't tell anybody. Yep, there's some froth. I denatured it, but there you go. Now I have a protein solution. What I'm going to want to do is take some of this protein solution and some loading buffer and put it in another tube, which I forgot to go get ahead of time. And then you're going to use the itsy bitsy pipette tips to load these onto the gel. And there's a really good video showing you how to do this on the website. But loading buffer, the blue stuff, is glycerol, so it's heavier than water. So what you do is you pipette up some of it into your pipette, and then you put it very carefully right above the well and push down the thing. And because it's heavier than water, it'll fall to the bottom of the gel. And because it's blue, you can tell it's there. If you want to be very good at loading these things, here is a hint. First of all, you definitely want to brace it against your thumb like I'm doing. Notice I've got my thumb on the back of the gel. That helps me maneuver this to right above the well I want to fill. Also, remember that when you're using these quantitatively, you push it all the way down. But this is not a quantitative experiment. This is a qualitative experiment. You don't have to get every last little bit into the well. So if you only depress this to the first stop, you won't eject a bubble of air. If you eject a bubble of air, your solution could wash out of the well. If you don't eject a bubble of air, it'll just sink down and look very pretty. And so you look like a professional gel filler if you do that. So you should have 2 tubes here, half protein, half loading buffer. You really only need about 20 microliters, a teeny tiny amount, like, you know, that much. Hardly any at all, because these wells don't hold a very large volume. They only hold about 30 microliters total. And you only want a little at the bottom of the well. So I would make a couple of suggestions. Don't use 2 wells that are next to each other. You've got, I think, 10 to choose from, and you've got the standard and the 2 protein solutions. And so that's only 3 wells. You can easily leave empty wells between them. I'd also go load asymmetrically, like maybe use the second well and then skip 2 and then do protein, space, protein, and then 2 blank wells or something, and make a note of what you do. Because when you bring this back to lab next week and we stain it, you're gonna want to know what was in which well. All right, so don't forget, you're gonna dissolve the protein in water, and then the protein and loading buffer, 50-50, go into a second tube, and that's what you load on the gel, because you gotta have that heavy blue solution added so that it sinks and that you can see it. So once you have your wells loaded up, then you're gonna wanna put the lid on, and then, and only then, are you gonna plug this thing in. This is, again, really tough, red to red, black to black. You're gonna turn it on. It has a couple of on switches. There's an on switch there, and there's a rocker switch in the back. So you gotta hit the rocker switch in the back, then you hit the on switch on the front, and the lights light up. And then follow the instructions in the lab. It tells you how to set this and hit run. Once it starts running, give it about a minute or 2, and then look, and you should actually see bubbles rising inside the cassette. That tells you it's running. And after about 5 or 6 minutes, you'll notice that this blue solution that you've loaded into the well is creeping down the gel. Watch it until the blue solution is most of the way down the gel, but not all the way to the bottom. At least 2/3 to 3/4 of the way down the well. Then it's finished and you can turn the whole thing off. That's what you're trying to do. So once you get there, then I've got to show you how to take this thing apart. So we'll leave it running for a little while and we'll be back in a few minutes.